Welcome to Chapter 13, Part 3, Using Chemicals to Control Microorganisms. In this lecture, we'll cover chemical disinfectants, antiseptics, and sterilants, focusing on how they work, when they're used, and what their strengths and limitations are. Chemical control is essential in healthcare, food preparation, and laboratory settings. Pretty much anywhere microbial contamination must be minimized or eliminated. And no single agent works best for all microbes or surfaces. We really choose based on the target organisms, the material compatibility, safety, and cost. And we'll see later that germicides are categorized based on what they can kill. If they can kill pretty much everything, these will be high-level germicides. If they can destroy only very basic microbes like vegetative cells, we call those low-level germicides. Let's start with a group of chemicals known as phenolics. These are one of the oldest classes of disinfectants, and they work by disrupting the cell membrane and denaturing proteins, which leads to cell leakage and eventually cell death. Examples include phenol, which is shown at the bottom left. We have cresols, O-phenylphenol shown in the middle, and hexachlorophene shown on the right. So you can see some of these have one ring, so phenol has one ring, and some of them have two rings, like the middle and the far right one. When you have two rings, these are in the group bisphenols, and these are very closely related. So these examples are often found in hospital disinfectants and Lysol type cleaners. Triclosan is a bisphenol that was once common in antibacterial soaps, and what it did was inhibit fatty acid synthesis, making it more like an antibiotic than a general disinfectant. But since maybe the last almost 10 years, it's been banned in consumer washes due to environmental persistence. So it was found a lot in seawater, in human waste, and also because of bacterial resistance. Phenolics in general are stable and they're broad spectrum, but they can be toxic and irritating in concentrated forms. So the deadline that companies had to remove triclosan from their products was September 2017. This was for all over-the-counter consumer antiseptic wash products, things like liquid hand soap or bar soap or body wash. They could no longer have triclosan in them. But other products like toothpaste, consumer goods, plastics that have antimicrobial additives, in those triclosan can still be used. They're subject to other frameworks. This checkpoint asks, why is triclosan more like an antibiotic than a traditional disinfectant? And it's because triclosan actually targets a specific bacterial enzyme in the fatty acid synthesis pathway, which is different from ours. We don't have that. And because triclosan acts on a single molecular target, bacteria can develop genetic resistance, similar to what they do against antibiotics. This is in contrast to traditional disinfectants, which act more non-specifically. They usually affect multiple cell structures, like the cell membrane and proteins, making resistance much less likely. Some heavy metals are also antimicrobial. So heavy metals such as silver, copper, mercury, nickel, and zinc have long been known for their oligodynamic effect. What this means is that even tiny amounts can inhibit microbial growth. And how this works is by binding to sulfur groups in proteins, which inactivates enzymes, denatures proteins, and thus disrupts metabolism. So examples include silver, usually in the form of silver sulfadiazine that's used in burn creams or catheters. Copper is often used to line incubators or door handles to reduce contamination. And zinc chloride, for instance, can be added to mouthwash. But because these metals can accumulate in our tissues and can be toxic, their use in humans is limited today. And our book provides us several examples of heavy metals. We have maybe copper in fixtures like door handles, silver in our silverware like forks, copper-lined incubators, zinc chloride in our mouthwash, and this patient is suffering from argyria, which is when there is accumulation of silver in the tissues in the body. This checkpoint asks, why are many heavy metals both antimicrobial and toxic to humans. So heavy metal ions bind strongly to sulfur-containing amino acids and proteins, and that denatures enzymes and disrupts essential cellular functions in things like bacteria or fungi. But the same thing would happen in human cells. So this can cause toxicity when metals accumulate inside the body, and we call that bioaccumulation. Halogens are another group of chemicals that are antimicrobial. So this includes iodine, chlorine, and fluorine. These are strong oxidizing agents that damage cell components and macromolecules. Iodine is used in betadine, and it's a povidone iodine antiseptic for skin and surgical prep. Chlorine forms hypochlorous acid in water. This is the active ingredient in bleach and water disinfection systems. We have chloramines that are slower acting but longer lasting forms of chlorine, and it's ideal for drinking water maintenance. And we have fluoride. This is the ionized version of fluorine. This strengthens tooth enamel and also inhibits bacterial metabolism in dental care. Overall, halogens are inexpensive and broad spectrum, but they can be corrosive and sometimes inactivated by organic matter. What is a benefit of chloramine over hypochlorite for disinfecting? Chloramines release chlorine slowly and steadily, providing longer lasting disinfection. Overall, they're more stable. They produce fewer irritating fumes than hypochlorite like bleach. And chloramines are commonly used to maintain residual disinfectant levels in our drinking water. Next, we have alcohols, and these include ethanol or isopropanol, used typically at about 70% concentration. These kill microbes by denaturing proteins and disrupting membranes, making them effective against bacteria, fungi, and envelope viruses because we can disrupt that outer lipid layer or envelope of the virus. These are fast-acting, inexpensive, and they leave no residue, and this makes them ideal for hand sanitizers and skin disinfection before injections. If we look at the second line, or this middle line over here, true or false, 70% alcohol is more effective than 100% alcohol. It's actually true. And this is because water aids in protein denaturation. Here you would have about 30% water. So water will help in denaturing those proteins and also slows the evaporation time of the alcohol for better contact time with the microbe. However, alcohols don't kill endospores, and they still evaporate too quickly to achieve full sterilization. This checkpoint asks, name at least 3 advantages of alcohols as disinfectants. So 1, they are fast acting and they evaporate quickly without residue. 2, they're inexpensive and widely available. 3, they have pretty broad spectrum activity against vegetative bacteria, fungi, and enveloped viruses. And interestingly, remember they work better at 70% concentration than 100 because some water is needed for protein denaturation. For the second checkpoint here, it asks us to describe several specific applications of alcohols used in disinfectant products. So sometimes we use them for skin degerming before an injection. So that's usually through isopropyl alcohol or ethanol, which is ethyl alcohol swabs. We see them in hand sanitizers, and they might also be in tinctures containing alcohol mixed with other ingredients like iodine for an enhanced antimicrobial effect. And then sometimes we use them for surface disinfection as well in healthcare and lab settings. Our next group are surfactants, and these are compounds that lower surface tension to help remove microbes from surfaces. So these include soaps. Soaps are mild surfactants that clean by mechanical removal. So they're not true disinfectants, but they are good for degerming. And if we look at a generic picture of one of these soaps, you can see it's a salt, and it has both a hydrophilic end and also hydrophobic end. We also have quats. Quats is basically short for quaternary ammonium compounds. You may have heard of benzalkonium chloride maybe at work. These are cationic detergents that disrupt membranes, and they're active against bacteria, fungi, protozoa, and also enveloped viruses, but they're not good against endospores or non-enveloped viruses. Quats are found in mouthwashes, surface disinfectants, and skin antiseptics as well. They're non-toxic and they're pretty effective cleaners, but biofilm forming bacteria can sometimes resist these quats. Here we have some examples of quats with their cationic end. This is the water-loving positively charged portion, and then our non-polar ends. And these disrupt cell membranes. So they're effective again against bacteria, fungi, protozoa, and envelope viruses that have a lipid membrane, that lipid envelope. Again, they're not effective against endospores or non-envelope viruses that don't have a lipid envelope, that are just surrounded by a protein shell. Why are soaps not considered disinfectants? So soaps do not kill microbes. Instead, they really act as surfactants that emulsify oil. So they break these larger particles into smaller particles, and they remove microbes mechanically during rinsing. So there are degerming agents rather than being antimicrobial chemicals. Although some soaps might have included like bacteriostatic additives, but the soap itself is not a disinfectant. And here our book has a little hand washing recommendation from the CDC for the general public. Basic steps to washing your hands thoroughly. Remember, soap is going to act as a degerming agent, although in certain cases there might be antimicrobial chemicals added to the soap. So I think a lot of the things that we sometimes miss is washing the backs of the hands, between the fingers, or under the nails. Our next group of chemicals are the bisbiguanides, and this includes chlorhexidine, and another one the book mentions, alexidine. These are widely used in medical antisepsis, and they work by disrupting cell membranes. This increases the permeability of the cell and causes leakage of cytoplasmic contents to the environment. At low concentrations, chlorhexidine is actually bacteriostatic, and that'll stop growth. But at high concentrations, it becomes bactericidal because it'll make the cytoplasm congeal, and that will lead to cell death. Chlorhexidine is also persistent on the skin, so it's widely used in surgical scrubs. It's also used in oral rinses and pre-operative antisepsis. It is broad spectrum and low in toxicity, although it's not effective against endospores or some Mycobacterium species. It's also ineffective or not very good against non-envelope viruses because these have a pretty stable protein shell surrounding their nucleic acid. What two effects does chlorhexidine have on bacterial cells? So it was one, disrupt cell membranes, which increases permeability and leakage of cytoplasmic contents. And at higher concentrations, two, it causes cytoplasm to congeal. That means the cytoplasm will kind of coagulate or solidify because proteins and DNA will coagulate. This will lead to cell death. So again, overall, chlorhexidine is bacteriostatic at low levels and bactericidal at high levels or high concentrations. Our next group are alkylating agents. And alkylating agents are highly potent sterilants that work by adding alkyl groups to proteins and DNA. And this will irreversibly inactivate enzymes and nucleic acids, which leads to cell death. Examples include formaldehyde and glutaraldehyde. These are used for sterilizing heat-sensitive instruments, but also for preserving tissue samples. We also have ethylene oxide. This is a gaseous sterilant, so this is a gas that actually can penetrate packaging, making it ideal for catheters or plastic equipment. And these agents kill all microbes, even endospores, but ethylene oxide is toxic, it's irritating, and it's carcinogenic, so it's never used on living tissue. Ethylene oxide is also explosive, so it has to be used very carefully and under carefully controlled conditions. Here we can see guanine, one of our nucleotide bases, is being alkylated. So here it receives an alkyl group, and that results in binding with thymine instead of cytosine. So we have altered DNA structure there. And then here are examples of several different types of alkylating agents: formaldehyde, glutaraldehyde, ethylene oxide, etc. What chemical reaction do alkylating agents participate in? So alkylating agents replace hydrogen atoms with alkyl groups, and these occur on proteins or nucleic acids. This will inactivate the enzymes or inactivate DNA, leading to microbial death. For the second question, why are alkylating agents not used as antiseptics? It's because they're highly reactive and toxic to living human tissue. They can cause chemical burns, irritation, and they're carcinogenic. So they're really only suitable for equipment sterilization or preservation of tissues that are no longer alive, not for living tissue. All right, our next group of chemicals are the peroxygens. Peroxygens are strong oxidizing agents that can function as both disinfectants and antiseptics. And the most familiar one is probably hydrogen peroxide, H2O2, which we've used in the lab. This will actually release free radicals, unpaired electrons, that damage DNA, proteins, and lipids. And these peroxygens are effective against bacteria, fungi, viruses, and in higher concentrations, even endospores. So you need higher concentration, but also a long enough exposure time. However, microbes that have catalase, if you have catalase, or the microbe has peroxidase, these enzymes will break down hydrogen peroxide. We saw that in lab before. So if these enzymes, catalase or peroxidase, are present in the microbe, then it will allow the microbe to tolerate low concentrations of hydrogen peroxide because it'll break it down into water and oxygen. So we have to think about whether our microbes are going to be resistant to hydrogen peroxide at low concentrations. We also have gaseous hydrogen peroxide and peracetic acid. These can be used as sterilants for equipment and entire rooms. Peracetic acid is stronger and it's catalase resistant, and it breaks down into safe products, actually acetic acid and oxygen. Other examples of peroxygens include benzoyl peroxide, that's often used in acne treatment medications or face washes. Carbamide peroxide, we see that sometimes in toothpaste. And ozone gas, this is used for disinfection of air and water. At the top, we've seen this in lab already before. This is the chemical equation showing hydrogen peroxide being broken down to water and oxygen by the enzyme catalase. And below, we see an image of hydrogen peroxide in a bottle. Remember, these bottles are usually darkened or opaque because hydrogen peroxide is sensitive to sunlight. And here we have contact lens cleaner. So the strongest type of contact lens cleaner you can get is the one that contains hydrogen peroxide, and you'll have a catalyst, usually down here, that will neutralize the hydrogen peroxide overnight so you don't put it into your eye the next morning, which would harm your eye. This checkpoint asks, how do peroxides kill cells? So peroxides, like hydrogen peroxide, kill cells by producing reactive oxygen species. So these are things like unpaired electrons, radicals. These radicals will oxidize and damage proteins, lipids, DNA. This leads to cell death. And these peroxides are effective against a wide range of microbes, but catalase-positive organisms can sometimes neutralize the hydrogen peroxide. This next checkpoint question asks, why is the use of supercritical carbon dioxide gaining popularity for commercial and medical uses? So I didn't include this part in my slides because it's very rarely used. It's really niche, so it's not very widespread practice at all. But supercritical carbon dioxide in general, what it is it's carbon dioxide that is between a gas and liquid state. And at this supercritical state, it penetrates materials pretty easily. And it kills microbes by lowering intracellular pH. It's non-toxic, not flammable, and it doesn't leave any chemical residue, so it's pretty good for treating heat-sensitive instruments, implants, and food. Now let's switch to chemical food preservatives. So chemical food preservatives are compounds that inhibit microbial growth to prevent spoilage without changing the flavor of our food. And common preservatives, you've probably seen them in your food labels, include sorbic acid, benzoic acid, and propionic acid, along with their salts like potassium sorbate or sodium benzoate. And they work by inhibiting enzymes and lowering pH, which interferes with microbial metabolism. So sorbic acid targets enzymes in the citric acid cycle and also often the catalase peroxidase systems. Benzoic acid can decrease pH. It also blocks oxidative phosphorylation, and we commonly see it in things like fruit juice, jams, or sodas. Propionic acid is effective at slightly higher pH, and it can prevent mold in cheese, and it's also good as a preservative in baked goods. We have other preservatives as well. These include sulfur dioxide, which prevents browning and spoilage in dried fruit and wine. We have nitrates, which maintain meat color. They can also inhibit Clostridium botulinum, although when you heat nitrites, sometimes you can form a derivative called nitrosamine, and that is actually carcinogenic when heated. We also have natural preservatives. Nisin is an antimicrobial peptide derived from Lactococcus lactis bacteria, and it inhibits cell wall synthesis in gram-positive bacteria. We also have natamycin. This is an antifungal agent from the bacterium Streptomyces natalensis, and it's used in cheeses and meats. What are the advantages and drawbacks of using sulfites and nitrites as food preservatives? So the advantages are they inhibit microbial growth, especially Clostridium botulinum. They can preserve the color of our food and also extend the shelf life. But drawbacks could be that they can cause allergic reactions in some people, or even potentially form harmful compounds like those nitrosamines I mentioned earlier during cooking or heating. So these food preservatives are used carefully in order to balance safety versus preservation effectiveness. And at the end of section 13.3, our book gives us a giant table that looks at all of the different chemical disinfectants we've talked about and lists them by chemical, their mode of action, and example uses. So take a look. I've broken the table down into 3 parts so we can fit it on the screen. So previously we saw the phenolics, metals, and halogens in the previous table. Now we have the alcohols, surfactants, bisbiguanides, and alkylating agents. We also have the peroxygens, our chemical food preservatives and natural food preservatives, and I briefly mentioned the supercritical gases, carbon dioxide, that's in between the liquid and gas state. And to recap 13.3, understand and compare various chemicals used to control microbial growth, including their uses, advantages, disadvantages, chemical structure, and mode of action. Again, you can use that previous table from the book, but in the next few slides, I've also compiled another table for you with these topics as headings. And here you can see I've added a few more headers compared to the previous table. For chemical structure, I'm not going to ask you to sketch it out, but you should have a general idea. Like earlier, we saw the single ring in our phenolics, whereas in the bisphenols, we had 2 rings. Heavy metals, I'm not going to ask you to sketch them out. We've heard of these before. Halogens, they're right next to the noble gases in the periodic table, like iodine, chlorine, and fluorine. Alcohols, we've seen this before. They have this hydroxyl group. Soaps, we saw soaps and quats. They have a polar end, a charged end, and then a long chain that's nonpolar. Bisbiguanides, don't worry about their structure, but we talked about chlorhexidine. And then alkylating agents add an alkyl group to DNA or proteins. For oxidizing agents, these peroxides, don't worry about the structure, although I would like you to still know that equation for hydrogen peroxide and catalase. We barely or briefly talked about carbon dioxide in its supercritical state between liquid and gas, and then don't worry about the structure of preservatives like sulfites or nitrites, but do know their mode of action, common uses, etc. And that takes us to the end of our third video. In the last section, 13.4, we're going to look at testing the effectiveness of different antiseptics and disinfectants.